Here is my Unit 4 video. So the first question was to draw the structure of the dipeptide THRLEU, the abbreviation, and then label the peptide bond, the N- nitrogen, and the C- for the carbon. And so here's my structure. And then you can tell I labeled this one, the N- for the nitrogen, I guess. Could move my arrow up. And then right here I put my peptide bond, and then over here is the C- for the carbon. The next question was to explain what type of forces stabilize each of the structural levels, and then point out how each of these structural levels are different. So first, I'm just going to explain what type of forces stabilize them, and then the back side of the card explains the differences. So for the primary structure, the peptide bonds are what stabilize the primary protein structure. And then for the secondary structure, it is the hydrogen bonds that stabilize the secondary structure. And then for the tertiary, are the hydrophobic interactions that stabilize this protein structure. And then for the quaternary are the hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bonds that all stabilize it. So that is that side of it. And then the back side explains the differences. So for the primary structure, it is strictly a structure of a sequence of amino acids that are creating a unique pattern. And then the secondary structure is different from the primary structure because it has a regular pattern or sequence of foldings on the polypeptide chain. And then for the tertiary, it has all three-dimensional shapes of one single polypeptide. And that also includes the interactions among the different groups, providing the functional form for the protein. And then the quaternary are the interactions among multiple peptide chains, functional protein complex. It's easier to tell the differences in a picture. Okay. And then the next question, 1C, was to describe two types of enzyme substrates interactions that lower activation energy, and then you can include drawings. So the first one was lock and key, and this one are the enzyme's active site is perfectly complementary to the substrate, and this one is like very specific, very detailed. And then another type of the enzyme substrate would be induced fit model, and this is where enzyme's active site changes depending on the substrate's binding. And then this active site adjusts to the shape, so it's a lot more flexible. Then the next question for 2A was to draw the structure of a nucleotide and then label the phosphate, sugar, and base. And so here's my drawing of the structure, and then I tried my best to circle the separate bases or the labels. So this first one over here was the phosphate, and then the second one was the sugar carbon, and then the last one was the base. There's my structure, nucleotide. And then for 2B, it was to contrast DNA and RNA by explaining how their molecular structures are different, and then to explain how their functions in the body are different. And so first, DNA has adenine, cytosine, guanine, and thymine, while RNA has the same, except it does not have thymine. So RNA has adenine, cytosine, and guanine, while DNA has all four. And then DNA contains deoxy, while RNA contains ribose. And then DNA is the long-term storage space for genetic information, while RNA is the translator or transportation for the genetic information from DNA to the ribosomes. And then that's how they're different in the body, performing different tasks. And then the next question, 2C, was to describe the structure of viruses and explain how viruses infect cells and replicate. So host cell nucleic acid flow. And so viruses attach to specific receptors on the cell surface, and they do that by using the protein spikes. And then I also included that viruses are infecting agents. And then for so part A to the last question was to define catabolism and anabolism. And so first up, catabolism is the breakdown of complex molecules into smaller ones, and catabolism also releases energy, while anabolism is the synthesis of complex molecules from simpler ones using energy. So catabolism releases energy, while synthesis uses energy. And then the next part was to name two types of chemical reactions that tend to be found in catabolic pathways, and then two types of chemical reactions that tend to be found in anabolic pathways. So the first question for the two types was oxidation and hydrolysis. And then for the second part, that was also two types of the two different reactions, was a reduction reaction and then a dehydration synthesis for the ones I found as examples. And then 3B was to draw a chemical equation showing the reaction, this one right here, and then to label the ATP and ADP as comparatively higher or lower in the energy, and explain whether the reaction is exergonic or endergonic. And so here's my chemical equation of this side and then this side. And the left side is actually going to be the higher side in energy. And then it's going to move down to a lower energy. And so you can, I included this, that ADP is lower in energy than the ATP. So higher side, lower side. And then this reaction is exergonic. The energy is being released. And then the last question, I think I have it as two sides. Yep. So the question was that explain how a starchy meal is digested in the body, and then be sure to describe the enzymes involved in the mouth and small intestine. So my answer would be that when consuming a starchy meal, the digestion process, let me restart this one. Okay, my answer was, when consuming a starchy meal, the digestion process begins in the mouth, and the saliva in the mouth then breaks down into polysaccharides, and then they get broken down into smaller molecules once the food is swallowed. And then, once the food is being swallowed, the enzymes in the small intestine break down the food, including enzymes like maltose, sucrose, and lactose, which then get split into monosaccharides such as glucose. And then they are then absorbed by the body and used as fuel. So that was my Unit 4 video. And those are all my questions and answers.